Today, we are learning about GRI outside of the brain, and we are joined by Dr. Dereka Boui, who's a professor at McGill University, and Dr. David Lynch, who's a professor of neurology at the University of Pennsylvania. And we're going to have Dr. Boui present first. So if Calliope, if you want to share Dr. Bowie's slides.
Okay, we're ready to go, Megan? Okay, great.
Yes, we are.
So welcome, everyone. I'm switched off my video because I'm in Scotland and I'm in the back of my parents' house trying to do this presentation. And so in the interest of trying to make sure that this runs smoothly, I've got my video off. So I guess everybody can hear me, Megan, right? Is that right? My audio's coming through. Right, great. Thanks. That's great. Okay, so thanks for the invitation. It's great to be back with everybody from CureGrin and to chat with them. We're going to be talking about inotropic glutamate receptors outside the brain, and Dr. Lynch has made some very interesting observations recently. And so my job here is to try and introduce a little bit of the topic so that he can then take you through the really nice work he's just published in the last year or so. So on the first slide, it's just to show you where I work in Montreal in the biomedical complex. So in terms of inotropic glutamate receptors out the brain, so we can move to the next slide. Although inotropic glutamate receptors were cloned from the brain almost more than 30 years ago, it's been known for some time, and this is just some RNA-seq data that I got from some of the many different types of databases you can get online. It's been known for some time, and if you look at the top of the slide, you can see that there's transcripts of different types of inotropic glutamate receptor subunits. In this case, GLU1 subunit, which is a common subunit that makes up the Nmd receptors. And if anyone's familiar with neuroanatomy, you'll see some of the different parts of the brain are identified. But you can also see that right on the left-hand side, there's adipose tissue, which is fat tissue, aorta, which is a major blood vessel. Of course, coming out the heart goes up and also down to your legs. It's also found in the blood. You can actually find transcripts in the blood. And then all the way to the extreme right, you can see that transcripts for GLUN1 has been found in urinary bladder, thyroid gland, skin, spleen, all over the place. And so people like me who have been working on inotropic glutamate receptors for a number of decades, we've been aware of this, and not many of us actually really tried to look at it. And one of the ways, I guess, in a simple-minded way, most people thought, well, maybe it was there and there were transcripts being made, but perhaps there weren't any proteins being made, and these inotropic glutamate receptors perhaps weren't really contributing to very much. That's obviously not true, and you're going to hear about that in the next presentation. But I think that's how a lot of people have been thinking about it over the last decades. And so I think it's a really interesting moment now to actually move forward and start thinking about what are these glutamate receptors doing that are sitting outside the brain. So before I get to that, what I'd like to do is just tell you about, just to remind you, because I know there's people on here who are invested in knowing about all the biology of inotropic glutamate receptors. What I thought I would do is just try to take you through just the canonical roles of inotropic glutamate receptors so you can see when Dr. Lynch speaks how his work really differs from how we think about inotropic glutamate receptors work in the brain. So we can move to the next slide, please. And so I hope everybody's seeing the next slide. So next slide shows a full-length cryo-EM structure of an AMPA receptor, and it's color-coded. So you can see that it's actually made up of 4 different subunits. In this case, they're actually all GLU-A2 subunits of the AMPA receptor. What's really remarkable about glutamate receptors, which I'm going to get into the next slide, is that although they actually fulfill very different types of functions in the brain, their basic architecture is basically the same. So what you can see is that structure, which is now published by Sasha Sobieski, who was working in Eric Kuo's lab. But what we know is that that basic structure is also found for all the different types of classes of glutamate receptor, which I've got listed here on the right-hand side, the NMDA type, inotropic glutamate receptors, the AMPA receptors, and also the kainate, and even the orphan class of inotropic glutamate receptors. They all have this same architecture. And so by that, we can move on to the next slide, please. So despite that, inotropic glutamate receptors, although they have this very similar architecture, they actually fulfill very different roles within the central nervous system. So I'm now still talking about these canonical roles of inotropic glutamate receptors. And so on the bottom part of the slide, unfortunately, I can't use my mouse because I don't think you can see my screen. But what you can see is the Nmd receptors are usually partnered up at glutamatergic synapses, which I'm going to tell you about in a few minutes. Then the Nmd receptors actually have this rather slow response to glutamate when it's released into synaptic cleft. And they bring a lot of calcium inside neurons, and they drive a whole bunch of very important processes that are important for learning and memory, for example, in the adult, but are actually fundamental for the development of the brain. So without Nmd receptors, our brain just doesn't develop properly. And of course, that makes a lot of sense for people who are listening here with single point mutations in Nmd receptors, and you can see how that can impact the development of the brain. The AMPA receptors, and I'm going to tell you about this a little bit more, partner up with Nmd receptors because it turns out that they're really vital to get the Nmd receptors functioning. And then another type of very important inotropic glutamate receptor that's found in the brain within this canonical role within synapses are Kinetic receptors. And what we've learned about Kinetic receptors, although there are a lot of similar properties to AMPA receptors, they seem to be really regulating the kind of traffic police of neural circuits that found on the post and presynaptic side, where they seem to be regulating the activity that's being driven by the AMPA and Nmd receptor partnership. So from that one common architectural design, you get these very, very distinct functions, which are really fundamental to brain function. So in addition to that, not surprisingly then, if we move now to the next slide, then the different types of inotropic glutamate receptors have been tied into different types of CNS disease. And what I have in this slide is just these three canonical inotropic glutamate receptors and how they've been identified. And this is a very, very limited number of the diseases that have been tied into, but tied into different things. So Nmd receptors for more than 30 years have been tied into stroke, so cerebral insult, also to Alzheimer's disease. AMPA receptors involved in a bunch of different things. Some of them include ALS or motor neuron disease. And they've also been tied into glioblastoma multi-form, which is this very aggressive brain cancer, which I'm going to talk about towards the end of my talk. And then finally, Kinetic receptors have been, a lot of companies have been interested in them because they've been tied into neuropathic pain. And then of course, because they regulate neuronal circuits, then have also been tied into maybe in the role in epilepsy and maybe being a target to control epileptic seizures. So moving on to the next slide. So despite these very diverse properties of these receptors, the canonical view in which we look at inotropic glutamate receptors is shown really summarized in this slide here. So on the left-hand side, you see a cryo-EM image of a typical glutamatergic synapse. The top part, which is labeled presynaptic, you can see there's all these little ping pong balls, if you like, which are vesicles filled with the neurotransmitter glutamate. And they're waiting to be released into that tiny little space between the presynaptic cell and the postsynaptic cell, where you've got this very dense staining in the cryo-EM image. And that's due to all these proteins, which are shown on the right-hand side in that schematic. And it's packed full of these different kinds of proteins. And if you look very carefully, you can see there's the AMPA receptors there, which are blue and on the left-hand side. Then, of course, the very important role of Nmd receptors there, okay? So what's important to appreciate is that they're not just sitting there alone in these synapses, but they're packed full of all these proteins. And that's why it's so difficult to try to understand and sort of delineate all the different kinds of properties of these proteins and how they interact with each other. But how we tend to think about it when we're often talking with patients groups and everyone like that is shown in the next slide. Where we distill everything down to really this very simple prototypical inotropic glutamate receptor. We have an AMPA receptor, which is usually co-expressed with an Nmd receptor. And the reason that they are actually partnered up with each other is because the AMPA receptor is fully functioning at synapses at rest, so you can actually record AMPA receptor activity. But it turns out that Nmd receptors are actually tonically blocked at the membrane potential, which cells sit at in the resting state of a cell. Then Nmd receptors are actually tonically blocked by magnesium. And so you need the AMPA receptor to actually cause a depolarization of the postsynaptic cell so that that magnesium block can be relieved in terms of the Nmd receptor. And then, as you can see with the arrow, they can then bring huge amounts of calcium inside the cell. And so that property brings about the sort of property that's been tied into coincidence detection for Nmd receptors. So that's a canonical way in which we like to think about different types of glutamate receptors. And of course, what we realize now that there's much more to these, much more to this than meets the eye. And so what I'm going to do in the next few slides before I finish is just tell you about a couple of these to try and set things up for Dr. Lynch's talk. So the first thing is some observations we've summarized. I'm not going to take you through any of our experimental data. I'm going to show you a couple of movies in a few seconds. And it's really this idea that the Nmd receptors in the neurons we've been looking at, we think this is probably something that's repeated all over the brain. So the Nmd receptor, if you look very carefully at the bottom left-hand side of this schematic, the Nmd receptor is kind of purple. So the Nmd receptor, by bringing calcium inside neurons, actually, in addition to regulating within the glutamate, it actually acts a bit like a conductor of an orchestra, and it regulates both sodium channels, which regulate the firing properties of neurons, so affects the excitability of them. So it makes the cells very excitable, but at the same time, and over a slightly slower time scale, if you look down at the bottom, you see that the calcium also regulates this thing called nNOS, and nNOS is neuronal nitric oxide synthase, and that produces nitric oxide and kicks on a whole bunch of biochemical events, and it causes over a slightly slower time scale an increase in the strength of inhibitory transmission. So what that means is the Nmd receptors drive the excitability of cells through these sodium channels, but over time, then they actually strengthens GABAergic transmission and brings everything back down again. So it's like a self-controlling kind of homeostatic mechanism. In addition to that, and this is what I'm going to show you a little slide of, is that the Nmd receptor, by working through nitric oxide synthase, maybe go back just to the previous slide just for a second. Working through nitric oxide synthase, you can see it produces this molecule called NO, which is nitric oxide. Nitric oxide is a gas, and many of you are familiar with it if you've been a kid, went to a dentist, then they often give you nitric oxide to put you under a sort of anesthetic state so that they can extract teeth or whatever. And so what that really does, the nitric oxide, is it just basically overwhelms the normal endogenous system and actually induces that state of anesthesia. So in this case, though, the nitric oxide is released locally and acts on local blood vessels to cause vasodilation, okay? And so that's very important because when you drive the excitability of the cell, then that cell needs a lot of energy coming to it. And one way to coordinate that activity, that energy-dependent activity, is then cause a local increase in blood flow into the brain. I'm going to show that in the next slide here. If you can move on to the next slide. Okay, so I'm going to set you up for this. So this is a recording which is done on a capillary in the cerebellum, which is shown here on the left-hand side. And what you're going to see is you're going to see two things. So because these are acutely isolated brain tissue, then the capillary doesn't have actually any resting tone to it. So what you're going to see is we're going to actually stimulate it so that the blood vessel vasoconstricts. But then when we stimulate with NMDA, what you'll see is you'll get this transient vasodilation. So you get this opening up again of the capillary, and you'll see some blood vessels, and then you'll see it vasoconstrict again. That means you'll get constrictors. So what I'm going to do is, I'm not sure if it's Megan's controlling this, but at the bottom end of that image, there should be a little arrow to click on the movie. If somebody could do that, just there you go. So what you'll see is, you'll see it now vasoconstricting. You see that? And there's up at the top, there's some blood vessels, and there's a pericyte, which is vasoconstricted. NMDA comes in, it vasodilates, and then it vasoconstricts again because the NMDA stimulation is gone. So maybe run it again, because it's pretty quick. So there's the vasoconstriction going up at the top. You see it? And then NMD receptors are being stimulated now. You get this vasodilation. You see it? And then causes remote constriction again. So that's going on all the time in the brain, every time you drive neuronal circuits. And in this case, and this is not what Dr. Lynch is going to talk about, he's going to talk about something different. But in our case, we're pretty sure this is mediated by neural Nmd receptor vasodilation. So this is on the right-hand side of the slide. You see that the amount of vasodilation under regular conditions, you can see vasodilation. But if you now use a toxin in the cerebellum or even in the cortex, destroys but eliminates all neuronal activity in these voltage-gated sodium channels, no longer see any dilation at all, showing that Nmd receptor stimulation is really mediated by neurons, and because Nmd receptors are actually sitting on the blood vessels or on non-neuronal tissue. Okay. So what I want to finish with, Megan, if I've got just a few minutes or so, is just to talk about something which is really an emerging and a very exciting idea. Maybe you can move on to the next slide. One more slide. Oh, sorry. Right. So this is, yeah, maybe just go back one slide, just because that video was so quick. No, go back. Go forward again. Sorry, I'm explaining this. Yeah. So this is just a snapshot of those images. So what you can see on the left-hand side is just baseline conditions, and where the yellow is identifying a parasite. So I'm not sure how nice the image will be with you, since we've got a kind of poor connection. But then when you put this thromboxane A2 agonist, so that's a molecule that would normally be released, let's say, by platelets when these damage the blood vessels, you get vasoconstriction, but that constriction is around the parasite you actually see here. And then when Nmd receptors are being stimulated, which is on the right-hand side image, you can see you get this opening up of the parasite. So this, let's call it vasodilation. It's not exactly the blood vessel per se, but anyway, you get this opening up the capillary. Okay, so that's just a snapshot of the movie I showed. So yeah, now we can go on to the last slide. Sorry about that for the confusion. So what I just want to talk about is 3 studies that came in Nature. It was about a year or so ago, which is really exciting in the context of this brain cancer. So it's glioblastoma multiform, which is a very aggressive brain cancer. And usually once someone's diagnosed with it, they don't have a very long time to live. 6 to 12 months, I think, is the number I've heard in the past. What's been known is that the progression of cancer is often driven by neuronal activity. That's something that was known. But what we understood is, how does that work? And what we know is it's a cancer mediated not by neuronal cells, by neuronal cells, so glial cells. And it turns out glial cells also express inotropic glutamate receptors. And so there's different studies here. The top 2, well, all 3, the top 2 really focus on the fact that the glial neurons within the brain actually start to form these kind of pseudo synapses. So it's a new kind of synapse, if you between the neuron and the glial, that then, with the glutamate being released from the neuron, actually drives proliferation of these cancerous cells, and that actually drives the progression of the tumor. So that's really the 2 bottom studies, one from an old friend of mine, Thomas Coomer, and then the top study, I think, is really interesting, because what they did is they actually started to look at a whole bunch of different types of cancers, because one of the biggest challenges in treating cancer is when you get metastasis. So when the cancer that, let's say, originates in so breast cancer, but then it finds itself in the brain and then starts to spread in the brain. And when they looked at different transcripts of breast cancer, then they found very high amounts of 2B, the particular transcript, which is a Nmd receptor subunit. And what they found is that these cancerous cells claim themselves into the brain, same thing that these studies were showing. So even though the cancer starts on breast tissue, it actually makes its way, breaks it off from the breast, breast gets bloodstream, and then actually starts to form these kind of synapse-like structures between neurons and glial cells, and then drives this very aggressive brain cancer, which is very deadly to patients and very bad to treat. So what's really clear is that these canonical views of how we've thought about inotropic glutamate receptors, there's much more to it than that. There's other things going on. I think in the future, by looking outside the brain and looking at what inotropic glutamate receptors might be doing, there, hear more about this from Dr. Lynch. And there's going to be some other things that we're going to, new kinds of biology we're going to understand, and it may lead to new kinds of treatments, and perhaps it may even tie into some of the symptoms that many people are talking about in what we're talking. So that's really it. The last slide is, I think, me. And I'll be happy to take any questions. I hope I haven't taken up too much time. Yeah.
Thank you so much, Dr. Bowie. So now we're going to turn it over to Dr. Lynch. And if anybody has any questions, you can feel free to put them in the chat and we will answer them at the end of the session.
Okay, so hi, thank you for inviting me. Let's see how readily I can do the share screen here. If I do this fast, it's a sad comment on how much we've been doing this in the pandemic world. Okay, it looks like I'm doing okay here, except for one thing, I need to go all the way back up to the top. There. And can you all see what I have?
Yes.
Very good. So thank you for giving me a chance to talk today. It's been a while since I talked about Nmda receptors. If that comes up later, we can talk a little bit about that. Today we're going to talk about some work that we did in association with Doug Seen, Ray Panettiere, and the various individuals in their lab on looking at Nmda receptors in the lung, and specifically in pulmonary vasculature. Now, I must admit, Doug Seen is an old friend of mine. He's a hematologist at the University of Pennsylvania. Ray Panettiere was one of my attendings when I was an intern 35 years ago in medicine. He's a pulmonary specialist. So they come to this from a different, entirely different perspective than what I, as a lifelong neuroscientist, might have. To remind you briefly of what Derek was telling you about, remember that Nmda receptors in the brain, without going into detail, occur as in the context of a component of cell biology with presynaptic membranes, postsynaptic membranes, and glial cells modulating the excitatory transmission. They have this prototypic paradigm of cell biological structure. In addition, we have Nmda receptors and other receptors that are made as specific components of multimeric molecules, where we know the molecular biological contents to them, of Nmda not mixing with AMPA, not mixing with kainate, and certainly not mixing with metabotrophic within a single molecule. You'll notice that I've been in this field long enough where I haven't even put NR3 on this slide, which I believe we designed this about 1995 or so. So there's a very specific molecular biological structure to things in the brain. We would guess that if you go to the periphery, that same structure ought to be maintained, and the lack of evidence on components of this structure has been a reason that many of us have been skeptical that Nmda receptors exist to a large degree outside the nervous system. So what actually is an Nmda receptor? It can, in its prototypic form, it contains two NR1 subunits and two NR2 subunits. That's its molecular structure. But the other thing to think about it is an ion channel receptor. And for it to actually be active, three events must occur. You have to have binding of two separate agonists, glutamate, which is regulated at the synapse, glycine, which is again regulated at the synapse, and you have to have removal of the internal magnesium block, which comes from depolarization. So now that works well at a synapse, but if you go to the periphery, glutamate is everywhere. Glycine is thought to be everywhere, and there are alternative agonists for each of these sites. And magnesium's a high concentration everywhere. Cells are sufficiently depolarized peripherally, or that magnesium block is at least leaky or partially removed before you start. So we may be able to find the molecular structures of an Nmda receptor outside the central nervous system, but exactly how it's going to be regulated in function is something which is going to, in function, is going to be something which is a lot more complicated. And then if these things all occur, calcium goes through, and calcium is regulated everywhere. So there have long been evidence of Nmda receptors in the gastrointestinal tract. I'm actually an author at a time when I'm an early postdoc in 1993 with Dave Pleasure and Delma Broussard, finding the molecular biological components of Nmda receptor 1 in the gastrointestinal tract, where they're presumptively in the enteric nervous system. So that's cheating. That's nervous system still. There have been a lot of reports of NR1 subunits, in particular in the heart. You know, that's always been there. I've never had much of an opinion on it. And then as you see from Dr. Bowie's presentation, it's capable of regulating vascular tone, but that's actually acting from the central nervous system. So it's not as if the Nmda receptors are in cells outside the central nervous system. Dr. Panettiere and Dr. Seen came to us almost 10 years ago now with evidence from the pulmonary vascular literature that Nmda receptor antagonists could change the constriction of pulmonary blood vessels. The first instinct is artifact. But since they were all friends and they had some reasonable hypotheses, we elected to go through and try and explore this a little bit more systematically to get where we ended up. So this is where I've clicked off here somewhere. There we go. Done largely by one of my fellows, Yina Dong, who remains with me today. I'm going to move this here. The first thing we did is try and find the subunits of the Nmda receptor by RT-PCR. So we're going to look and try and amplify for the presence of mRNA. This is our control panel from brain. Primers designed for each subunit, and you can see that we can amplify for NR1, NR2A as we call it. Other people would call it GRIN2A. Again, I'm from the old era. NR2B, 2C, and 2D. When you take human pulmonary vascular vessels and amplify, you are able to see those same bands, each of the appropriate molecular weight, with the exception of NR2B. It is selective, so you would suspect what happens is in pulmonary vasculature, it's there, but it's utilizing a different splice variant with a slightly different PCR product length. Let's see if I can get to move forward. You want to go? There we are. My computer. The next thing we did would take selective antibodies for each one of the subunits and see if we can find them. PCAM is a marker of the endothelial cell lining for these various blood vessels. So that allows you to show where the endothelial cell layer is. There is some literature suggesting the presence of NR1 subunits alone in alveolar cells, the final air sacs, but there's no evidence suggesting that they're Nmda receptors on the endothelial cells of pulmonary blood vessels. So here we're using peroxidase-based detection. So this is an antibody to gluin-1, 2A, 2B, 2C, 2D, and this is the second antibody alone control to show that this is a selective reaction product. And what you see is a very almost linear brown staining in this layer here, which is inside the endothelia. So it's in the medial layer of the so-called medial layer of the blood vessel, where you have the SMC, the smooth muscle cells. Remember what smooth muscle cells do in a pulmonary artery is that they contract and regulate the luminal diameter. So here's the lumen here. And you see for each one of the subunits, perhaps strongest for 2D, that's probably the basis of the antibody, you see localization of selective reaction product and what appears to be the smooth muscle cells within these pulmonary blood vessels. Okay, let's see if we can move forward here. There we are. So now we've done something a little different. Victoria Stepanova, assistant professor in Dr. Seens's group, has dissected those pulmonary blood vessels and grown them in tissue culture so that you're growing under this situation. You're growing the smooth muscle. Oops, it went forward. Let's go back. Come on. There. You're growing the smooth muscle cells in culture. And again, so there's nothing else here besides smooth muscle cells at this point. Green is our reaction product here, and for gluin-1, 2A, 2B, 2C, and 2D, you can see reaction product immunofluorescence showing that you do indeed have Nmda receptors in these cultured cells. This is our no primary antibody control in the middle. Second antibody alone. This is counterstained with DAPI to show you where the cell bodies are, and the merge of the two. So these smooth muscle cells are making, by immunocytochemistry, each in our subunit receptors. You could also try and look by Western blot, but if you look by Western blot, looking to get the size of these receptors, you actually won't find anything because the protein is in so low abundance that you see only non-specific binding. What you have to do instead is to immunoprecipitate. You're allowed to concentrate a lot more tissue with that antibody. Our control in the first lane is hippocampal neurons grown in culture, so we know they're going to have an Nmda receptor. And in the third lane, you have the immunoprecipitation of smooth muscle cells in culture looking for your receptor. So yes, there is an appropriate size band for NR1, a very faint but appropriate size band for NR2A, GLUN2A. Again, for NR2B, partially proteolized one there. NR2C, again, and even NR2D. So you can detect them by by immunoblotting, and they are the same molecular weight, thus enhancing your idea that this is truly an NMDA receptor. You could also assess, well, are these receptors on the cell surface? So what we've done here is it happens to be the GLUN2B antibody and GLUN2D antibodies recognize extracellular epitopes, so you can simply leave the cells unlysed, so they're unpermeabilized, and you can allow the antibody to react, and you can detect it on the surface in patches of the individual cells. So you know this is surface receptor. You can also sort them by fact sorting and sort for both GLUN1 and GLUN2B, and what you can find is there's a population of cells labeled with both, so that it means that both subunits are in the cell surface. Another way to prove that these are truly physiologically active receptors is to do electrophysiology, as Dr. Boui would be very familiar with. So this is from Robert Sue and Doug Coulter's lab. He's done a patch comp recording of some of these smooth muscle cells where he's applied 100 micrometer NMDA, presence of no magnesium. Glycine is already present there. There's your size bar at 40 picoamps, and you can see a downward current characteristic of an NMDA receptor in this model. Not a little bit noisy, not the largest in size that I've ever seen, but clearly there. The actual mean across 8 cells, if you look at the mean current, you see about 10 picoamps of current. That is a small current. You would go into brain and record 100 picoamps, if not occasionally nanoamps, if in some transfected cell models. So the size of these currents is very small, but they're very clearly there and pharmacologically appropriate. You can enhance it a little by looking at the area under the curve to look at the base of the amount of charge transferred. So these are NMDA receptors. They're on the cell surface and they are functional. Are there other components to glutamatergic transmission made in these smooth muscle cells? Because for something to be a quote transmitter, not only do you have to have receptors, but there has to be a selective synthesis mechanism, although glutamate itself is not so specific. There has to be inactivation mechanisms, and there have to be packaging mechanisms. This looks for one of the an enzyme called serine racemase by immunohistochemistry in these same cultures. Serine racemase produces D-serine, which in some regions of the brain is thought to be the agonist at the glycine side of the NMDA receptor, particularly in the forebrain. This is what you see out of neurons, and this is the serine racemase you can see out of pulmonary artery smooth muscle cells. So there's serine racemase there. They should be making D-serine. In addition, you can see that these cells actually have what's called V-glut, a packaging transporter for glutamatergic vesicles. So yes, they do have the machinery to create a sort of autocrine system of stimulation within these smooth muscle cells to turn on these NMDA receptors. Is there any pharmacological evidence? Well, this looks at human lung postmortem from donors, where the pulmonary artery has been made to vasoconstrict during application of an adrenergic agent and phenylephrine. And what you see on here is the dose response curve going to maximal vasoconstriction. You can see that as you go to hypoxic conditions, it shifts the curve a little bit to the right. If you go to hypoxic and add NMDA receptor, it shifts it relatively far to the right. So if you look at the total amount of vasoconstriction, you can see that there's a big difference between the hypoxic and the vehicle treated with MK01. So MK01 blocks the vasoconstriction, so NMDA receptors are leading to vasodilation. They have to get this to an opposition to the effects of phenylephrine. There's no change in the actual EC50, the potency, and no change in the total amount. It's simply the amount of vasoconstriction at a particular MK01 dose. You could also do the same thing in mouse pulmonary artery vessels using not phenylephrine, but endothelin to cause vasoconstriction. And again, application of MK801 blunts the endothelium-dependent vasoconstriction. So you block it and it's vasodilating. So that means the endogenous lung smooth muscle Nmda receptors will be causing vasoconstriction, sort of the opposite response to what is proposed by neuronal control. This looks at the total Emax effect, and there is some shift in the potency with either MK801 or an APV, AP5, a glutamate site antagonist of the Nmda receptor. And the total area under the curve is also decreased, although the curves don't go all the way out. So again, these are functional Nmda receptors acting on the process of vasoconstriction of the pulmonary artery, which is what they do. Now, that was actually Doug and Ray's original interest, is what these might do in pulmonary emboli and things which alter vascular tone in the pulmonary artery. And that's where they would be looked to going forward in the area of clinical therapy. So what does this all mean? It means there's certainly Nmda receptors on the pulmonary surface of pulmonary artery smooth muscle cells. They are functional and they modulate vasoconstriction. Notice I did not say that they make it vasoconstrict. They modulate or vasodilate. They change what happens with other modulators. So that their absolute necessity is not clear at this point. This may truly be an endogenous signaling mechanism, not so much neuronal, but more likely autocrine, smooth muscle cell to smooth muscle cell, so that they all act together. Because there are no real nerves in this region. So you get the idea that it's more of an autocrine system rather than a neurocrine or a neuronal-based system. Is this important in vivo? Now, that's a really good question. There certainly have been lots of effects peripherally of Nmda receptor antagonists in stroke trials and things like that. They've always been thought to be mediated by actions in the brain, actions at brain vascular interfaces. No one's ever talked about the possibility that they might be relevant peripherally. I think it remains an open question, which is, unfortunately, I will tell you, hard to study. Why would I say it's hard to study? Well, these 10 slides that I put together here took a full 5 years of work by 2 people full time. When the receptor levels are this small, it's not surprising that you miss them at first pass or second pass or third pass, and it means they're quite difficult to study. Now knowing what to look for, it's a more doable event, but it is quite tough to say how much this means in isolation. I think that's a question for the future that I hope a variety of us will be able to move forward on. The most important thing I also have to say, though, is to thank all my collaborators. The work from my lab was done entirely by Yina Dong. She's been a postdoc with me on and off for about 20 years, and she's actually joining us on faculty soon. My close collaborators on this are my good friend Doug Seens, who also was one of my attendings when I was an intern, and his research assistant professor, Victoria Stepanova, Ray Panettiere, who is actually no longer at the University of Pennsylvania, a wonderful postdoc, Robert Sue, from Doug Coulter's lab, who I've worked with on various projects over the past 20 years, and then Doug himself is always great to help me with electrophysiology. So I thank you for your attention, and I'll take any questions that you have.
Wonderful. Thank you, Dr. Lynch. That was fascinating. We do have a question in the chat. Do you think the high glutamate concentration in the periphery could account for the low prevalence of Nmda receptor subunits in peripheral tissue? Could it be that these cells don't require that many Nmda receptors due to the high concentration of glutamate in the periphery?
So I think the high concentration in the periphery means that we have to sort of stop and think. Because tonically, these ought to be activated all the time because there's glycine around too. There's no real magnesium block. These should be tonically activated. Is it possible that during other times when glutamate concentrations were to fall even a little because you're acting on desensitized receptors, you might see a change in the vascular tone as it falls, perhaps? Is it possible with what you might suggest that glutamate leads to the internalization of some of the receptors, and if they're tonically internalized? We didn't see any evidence for that, but we didn't look. And that's a much easier experiment to do in a neuron than to do at high concentrations of glutamate. The pulmonary artery smooth muscle cells grown in culture are nominally free glutamate media. However, if any of you have ever done tissue culture before, one of the things that many cells do is that they take glutamine from the media and dump it out as glutamate. So it just, you can make it nominally free, but it's not free for very long. It's hard to figure out exactly how the glutamate in the periphery would, whether it make these less active or tonically active all the time, and whether there's a packaging mechanism to overcome that. The presence of VGLUT1 is intriguing. I don't know what to do with it yet. The presence of D-serine is intriguing. I don't know what to do with it yet.
Wonderful. Thank you, Dr. Lynch. So I don't see any other questions currently open in the chat. If anyone has any, oh, looks like Dr. Bowie has a question.
Yeah, thanks, Megan. I've actually got a question for Dr. Lynch. Is my connection still good? Can you still hear me?
We can hear you.
Yeah, okay, great. I think it's really interesting, the observation you made, and I'm just reflecting on some observations that were done on neurons. So Pierre Paoletti in Paris many years ago showed that Nmd receptors are mechanosensitive, and of course blood vessels are very challenged or dynamic. And then Mike Salter, I think, who's actually in on this call right now as one of the participants, he showed probably sometime in the 90s or something like that, he showed an observation. I think it was potassium regulates Nmd receptor function. Of course, potassium regulates the dynamics of blood vessel reactivity. And so I'm wondering, do you think they could be the mechanosensitivity and levels of potassium could be regulating Nmd receptors? And we've been thinking about it in terms of neurons, but now it actually might be important in terms of blood vessels.
I think that's an intriguing idea. I see Mike, there was sodium regulating Nmda receptor currents. I'll also note in passing that Yina was a postdoc with Mike for a few years in the middle for tours of duty in my lab. In any case, I think mechanosense is an interesting idea. We have a group at the University of Pennsylvania who has studied that in the context of head trauma, and whether head trauma and its exacerbation is a form of mechanosensitive excitotoxicity. I think that's an interesting idea. Now, to know that, we would have to go to the smooth muscle cells and know the orientation of them on the membrane, whether it is on the luminal side, the non-luminal side, or at the junctions between them. They are frequently coupled by gap junctions, so they would have the ability to pass things there as well. So I think that's an interesting idea. I have to think about it.
Okay, sure, thanks.
Another possibility, Mike says, is non-ionotropic effects of NMDARs. Ever consider those, David? Well, yes, since you helped train her, this is tough tissue to work with. There is the suggestion that the C-terminus may leave the Nmda receptor in these cells, and a portion by antibody will show up in the nucleus. Now, as you know, that is a hugely problematic experiment to stake your life on, because the antibodies are not particularly good and the number of cross-reacting proteins is huge. We have talked about whether that is the role, and I've talked about it in the nervous system as well. If you think about the C-terminus of the NR2 subunits, whether the messenger side in the C-terminal region is actually the big part. So I think that's another interesting idea. And these distally sized currents are only part of it. They're just there enough to activate whatever process is carried out by the C-terminus. I think it's another interesting idea.
Terrific. Well, thank you both for your time. We greatly appreciate having Dr. Bowie and Dr. Lynch join us for this wonderful session on GRI outside of the brain. You can join us next for a session on beyond seizures, or also our GRI medicine cabinet concurrent session. So you can click on the concurrent sessions in the Excel events and join there. So thank you all so much for your time. We greatly appreciate it.
Thanks.
Thanks, Megan.